Hello, everyone. We'll get started now since we're at 6:30 p.m. So I want to welcome you to tonight's mini med school lecture series. We're pleased to host a presentation and discussion this evening on the topic of the medicinal power of your microbiome. Before we get started, I'll just cover a few housekeeping items. So there will be two parts to tonight's program. First, each of our presenters will speak for approximately 15 minutes, and then we will open the floor to audience questions. Following the Q&A session, there will be an informal lecture or an informal reception for those of you here in person. You're welcome to ask your questions at any time during the presentations using Slido. To access Slido, you can scan the QR code that's up on screen right now, or manually enter the event code Snyder MMS into the participant field on the Slido website to post your question. Questions submitted to Slido will be viewable to all audience members, and participants can upvote the questions they would most like to see asked. Now on to more specific housekeeping items. For those of you that have joined us online, if you lose your internet connection and drop out, you can return to this session using the same Zoom link you used to join us now. If you experience any technical issues, such as you cannot see or hear the presenters, please email snyder@buxa.com and our team will do their best to support you. For those of you here in person, we now ask that you switch your cell phone to silent mode. Please note this event will be audio recorded and the recording will be posted on the Snyder Institute website and YouTube channel. Private recordings are not permitted. To begin tonight's program, I'd like to introduce our MC, Dr. Derek McCoy, who is the director of the Snyder Institute and a professor in the Department of Physiology and Pharmacology. In addition to his faculty and directorship appointments, Dr. McCoy also holds the Lloyd Sutherland Professorship in Inflammatory Bowel Disease and Gastrointestinal Research. Dr. McCoy's research focuses on the regulation of intestinal flora and function in health and disease with a particular interest in inflammatory bowel disease. Welcome, Dr. McCoy.
Thank you, Hannah. It's a wonderful privilege to be here as the director of the Institute to welcome you to Mini Med School tonight, where we'll update you on some of the very exciting things we're doing with microbial therapeutics and living medicines. And to start off in a good way, I'll take this opportunity to recognize that the University and City of Calgary are on the traditional lands of the Indigenous people of Southern Alberta. And it is a privilege to be here and to recognize the knowledge keepers and the keepers of the land that have looked after this land for millennia. In the spirit of the land acknowledgement, I use this as a time to reflect on what has happened in the past, to make some effort to learn about our history, our present, and think about what it may mean to me personally as we move through into the future, and to act in the spirit of truth and reconciliation. So with that, the Snyder Institute for Chronic Diseases is a group of about 450 investigators, faculty members, staff scientists, and trainees, and all with the common goal of making groundbreaking discoveries leading to better treatments for chronic and infectious disease and improving healthcare. That's the primary goal. Our work will drive improvements in healthcare. And ultimately, cures for these conditions, which are often idiopathic and very difficult to cure. We bring together MD scientists, PhD scientists to tackle issues that are relevant and to drive improved clinical outcomes. We have world-renowned researchers working in gut, lung, pancreas, liver, kidney, and a variety of infectious diseases and other conditions. And while we do have a focus on education and community outreach, we are primarily driven to be a research-intensive institute. And with that, the goal is to improve patient care. This research is funded by competitive peer-reviewed grants. And these are very competitive, and many excellent grants are not funded really based on budget. We are leaving excellent grants unfunded at the federal and provincial level. And of course, philanthropy has a huge role to play and is critical in what we do. As a couple of examples of advances that have really made a difference and will make a difference as we move forward, one of our investigators, I'm sure you've heard of C. difficile infections and FMTs, that started here in Calgary and is now the acceptable form of treatment for C. difficile infection in patients infected with that organism. Another one of our investigators is running a longitudinal study, the Bloom study, that's looking at preterm infants, monitoring their microbiome, their bacteria over time, over the next 3-5 years and hopefully beyond, to try to understand our early life changes in the bacteria in the gut or the lung or the skin. Is that predisposing those individuals to any inflammatory disease or any chronic disease? And then within the last 2 to 3 years, we've really pushed quite hard in creating an organoid facility. So this is the ability to take cells from an individual or little pieces of tissue from an individual and culture that into a simplified organ. And that organoid then allows us to test the impact of bacteria from that same individual or a different individual, and to test drugs on those tissues before they might be prescribed to try to optimize personalized treatment using these organoids. We have world-class research facilities and talent, and these are what enable our discoveries. Indeed, as far as I know, we are the only institution in Canada that has put together a world-class organoid facility and a world-class imaging facility. So we can understand what's going on in those tissues as they're exposed to bacteria, looking at how these bacteria and drugs affect those tissues and image that in real time. And that's unique across Canada. In 2017, just before 2017, there was a real push to drive into understanding the role of bacteria in health and disease. And we managed to recruit Professor McCoy, who will speak to you in a moment. And in 2017, Kathy and her team opened the International Microbiome Center. This is a germ-free facility. This is the largest facility in North America, and I dare say it's the largest germ-free facility in the world. And this allows us to really look at that microbiome, those bacteria, that microbiota, to really understand how these bacteria interact with tissues, whether they're mouse or human. So that is the topic of tonight's presentation, to look at the development of microbial therapeutics. And you will hear from their two speakers our achievements and our potential to really revolutionize therapy for a variety of chronic and infectious diseases by harnessing the power of microbial therapeutics. Dr. McCoy and Dr. McDonald will tell you of some of their really exciting discoveries. So with that, I'm going to hand over to Dr. Kathy McCoy. Kathy is a professor in the Department of Physiology and Pharmacology in the Cumming School of Medicine and a primary member in the Snyder Institute. A world-renowned scientist. She's an immunologist by training. She's been leading the field in microbiome research for a number of years now, and really is a global leader in that area with respect to the microbiome, bacteria in health and in disease, as it affects us from early birth right through to aging. So with that, Kathy, the floor is yours. Welcome.
Good evening, everybody. Can everybody hear me through the lapel mic? Okay. If not, just raise your hand and I'll go to the other mic. So it's really a pleasure. Yeah, you can't hear? Okay. I'll go over here then. Is that better? Okay. Okay. So good evening. It's really a pleasure to be here, and I have to tell you, I really love talking about this research here because it excites me every single day. It excites me of the medicinal power of your microbiome. What we've achieved here in the past 10 years, I find really exciting. And, the future prospects, I think, are even more exciting. So in my sort of 15 minutes, what I'm going to do is I'm going to sort of give you, again, an overview about what the human microbiome is, why we believe it is so important. I'm going to give you sort of a snapshot of some of the stuff we've done here in the past 10 years. And then I hope to really excite you about what is our next endeavor here at the University of Calgary. So having said that, I want to remind everybody here, I think you probably all know that every single one of us are colonized by trillions, 400 trillion bacteria that live in your body. And so that means that every single mucosal or barrier surface in your body is colonized with a community of microbes. So if you think about any part of your body that is ever exposed to the external environment, I can guarantee you that there will be microbes that are living on that. And so barrier surfaces are like the skin, and anything that is exposed. So your lungs and especially your guts. So they're all colonized by microbes. And although our research has been really heavily focused on bacteria, because they are the predominant biomass of microbes that live on you, we have to remember that there are other microbes that live on you, and that includes viruses, fungi, protozoa. Even there are viruses called bacteriophage that infect the bacteria that live on you. Even our microbiome has a microbiome. So it's sort of a very powerful system. And so the composition and the diversity and the function of those bacteria that live in and on your body is really influenced by their environment. So a bacteria or bacterial community that lives on your skin looks very different to that lives in your gut, because those environments are very different in terms of oxygen level, pH, temperature, and the nutrients that are available for them. So it's important that we look at the site that we're studying. And another thing that we've come to realize over the past several years is that all of us are unique. We all have our own individual microbiome. And so if you look at it at a very high level, there are 55 different bacterial phyla in our world. There are only like majority of 4 phyla, go up to 12 phyla that can inhabit our body. So it's still sort of a host system or environment that dictates what bacteria can live there. But when we start to look more and more detail, then it becomes very individualized. And that's because it's influenced by the environment. And while all bacteria, all mucosal and barrier sites on your body are colonized, we typically study the gut, because the gut is really home to the vast majority of bacteria that are in your body. So if we talk about the development of the human microbiome, we believe that in utero, when a fetus is developing, we believe that is in a sterile environment. We don't believe there are bona fide microbial communities that are colonizing the fetus while in utero. But we do know that the development of that fetus, even in utero, when they're not colonized with their own microbes, is still influenced by the microbiome, because they're receiving signals from the maternal microbiome. And then that baby is born into our microbial world. And during the birthing process, that baby becomes colonized with their own communities of microbes. And then that goes through this really dynamic phase of colonization in the early period of life. And I just want to highlight that this period of time in the first 3 years of life is a very dynamic phase of microbial colonization. It goes through successive waves. Bacteria come in, change the environment, new bacteria come in. So it's always changing. That's the exact same time that a baby's immune system is still developing. It's also the exact same time that a baby's brain is developing. And this is a really important time in development where there is continual crosstalk between the microbiome and that developing immune system and brain. Now, that whole composition of microbiome is heavily influenced by many different factors: maternal factors, the mode of birth, if that baby is breastfed or formula fed or a combination, the lifetime use of antibiotics, your infectious history, the diet throughout your entire life, and many other things. A little bit about our own genetics, how active you are, where you live, if you smoke, who you live with, how much exposure you have to animals, to pets, to cows and horses, your stress, your medication. These are all factors that are going to change and modulate your microbiome. So why do we care so much about the microbiome? It's on the outside of our body. It's not inside our bodies, but it's in your intestines and your lungs, and it's separated by the inside of your body by these barrier surfaces. Well, we're super interested in this because we now know that the microbiome has an impact on nearly every host system that we have. And that is really how it's very important to educate the immune system. It's important in development and all these different factors. So it really is one of the most important factors that is going to educate and determine health and homeostasis and an impact on disease. And so for examples of these sort of systems, we know that the microbiome in early life is very critical to educate and influence the development of the immune system. And if things go wrong during that period of time, then we know that alterations in the microbiota during that really critical window in early life, it can increase susceptibility to immune-mediated diseases later in life. And that includes allergic diseases like asthma, food allergy, also autoimmunity like type 1 diabetes, multiple sclerosis, and these different diseases. But it's not just this early life period, although it's really critical. The microbiome still regulates and influences the function of your immune system all through life. And this is really important if you think about any immune response that you have, your response to vaccination, your response to infection, your response to any kind of immunotherapy you may receive. Microbiome also modulates brain development. So it modulates neural development in early life. And so if you think about that, it also modulates function. So that means that the microbiome can have an impact on both neurodevelopment and potentially neurodevelopmental disorders, on depression, on behavior, and also on the neurodegenerative behaviors or disorders. And it also modulates your digestion and metabolism. So microbiome helps to regulate your metabolic rate. It regulates fat stores. So we know the microbiome can play a role in if you're lean or obesity. So these are huge factors that the microbiome modulates in every host system. So I'm going to just sort of hone in on one of these things just because I don't have time. They've only given me 15 minutes, so I don't have time to tell you about everything. So I'm just going to focus on one thing that my lab has been studying now that I think is really exciting, and that's cancer. Now, we know that the microbiome modulates cancer and the response to cancer immunotherapy. Now, this figure is taken from basically the hallmarks of cancer. And since 2001, I think, they've been publishing these hallmarks of cancer, which just says, what are the factors that influence cancer development, progression, response to therapies? And it started off with sort of 6 factors. It was updated in 2011 with more factors. And in 2022, it was updated again. And it's the first time that it was realized and really documented that microbiome is one of those hallmarks of cancer. And we know, and we've known for a long time, that the microbiome or microbiota that you have can modulate your development of cancer, your progression of cancer, and also how well you respond to treatment. Now, there are some ways that the microbiome can have direct effects on a tumor, on cancer growth, but the majority of the way the microbiome modulates cancer is through its modulation of the immune system. And I know I talk about the immune system a lot because I'm biased, I'm an immunologist, but this is really one of the major ways that the microbiome shapes host homeostasis and disease. And you know, this is really exciting for us because our genes are not modifiable. We're born with a certain genetic makeup and we're stuck with that for the rest of our lives. But our microbiome is influenced by the environment, and microbiome, therefore, is potentially a modifiable factor. And that means maybe we can target the microbiome or harness the microbiome to modulate development of cancer, progression of cancer, comorbidities of cancer, and response to therapy. So we now know there are many cancers that are associated with being pro-tumorigenic, but we also know there's many microbes that are anti-tumorigenic, and they might be having effects directly on the tumors, and a lot of them are having an effect on the immune system and modulating the immunotherapy response. And to give you a little bit more information about this, a few years ago now, it was published that one really groundbreaking form of cancer immunotherapy is called immune checkpoint therapy. The 2 scientists that discovered this won the Nobel Prize a few years ago, and it's really revolutionized the treatment of cancer, especially melanoma and lung cancer. And this is basically harnessing the immune system. You give an antibody to take the brakes off the immune system, activate the immune system again to recognize the cancer and fight it off. Now, if you use this immunotherapy and you look in a mouse model, so a germ-free mouse that has no microbes at all, that immunotherapy doesn't work, has zero efficacy. And that tells you absolutely need a microbiome for that immunotherapy that these two scientists won a Nobel Prize for. So the microbiome is important for that. They also identified some bacteria that seem to be associated with a better response to this immune checkpoint therapy. We also know that people that unfortunately have had to take antibiotics prior, 3 to 6 months prior to getting this therapy, it's really associated with a worse response to the therapy. We can sequence the microbiome between patients that respond well or those that don't respond well, and the microbiome looks different. And they also did studies where they took the microbiome from a patient that had melanoma and had responded to the therapy. And they put that into this germ-free animal model, gave them the same tumor and the same immunotherapy, and that mouse responded. But if they take the microbiome from a person that did not respond, put it into the same germ-free mouse, give it the same tumor, the same therapy, that mouse does not respond. Now, this tells you that you can transfer the responsiveness to immunotherapy just by transferring the microbiome that the patient has, telling you how powerful the microbiome is, determining whether that therapy will work. And then people have also done fecal microbial transplantations. They've taken the fecal microbiota from patients that had melanoma that had responded to this immunotherapy and had a durable cure, and they put that into patients that had already failed this therapy. And in both of these studies, they were quite small studies. In both of them, they had patients that went on to have curable disease. And these had already failed the same therapy. So again, telling us that maybe microbiome can be used as an adjuvant therapy for cancer immunotherapy. So I hope I've excited you about the power of the microbiome. And now I want to sort of change gears a bit and tell you a little bit about us here at Calgary. So what have we done? What have we done in this field of microbiome research? So about 10 years ago now, it was really a focus here at the University of Calgary and in the Snyder Institute that we wanted to study the microbiome. So we asked the question, what do we need to do to make sure that here at the University of Calgary, we can do the most innovative, cutting edge, and translatable research on microbiome? So we looked around and we said, what do we need to build? Who do we need to recruit to make this happen? And so what we've done is now we've built the International Microbiome Center. And Derek introduced that a little bit just a few minutes ago. And we like to say that this is where cutting edge research meets comprehensive services. And so what we did is we had a very focused drive on basically building up the Microbiome Center. And together with the University of Calgary, the Cumming School of Medicine, and the Snyder Institute, we did a very focused recruitment drive to bring in researchers with world expertise in microbiome research. We also built up the infrastructure and the programs and platforms to be sure that all the researchers here can do really innovative research, because our goal is to harness the power of the microbiome. And so for an example, these are all the different integrated platforms that we've built, but just to highlight this to you, what we've done is, we talk to clinicians, and you're going to hear from Braedon later, and we're trying to convince them all, when you have a study, you should be collecting microbiome. Don't ignore it. It's not just waste. We're really interested in it. So we give them all of the protocols, convince them to collect microbiome in all their patient cohorts, and then we give them the right protocols, and we get them couriered to us at the Microbiome Center. We take those, and then we make sure that they're processed in the right way. Most of the bacteria in your gut don't like oxygen. Oxygen kills them. They need to be in an anaerobic environment where there's no oxygen. So we take them and we put them in these special anaerobic chambers where there's no oxygen, so that we can process those and make sure we store them so that we can do all the downstream analysis on that is going to tell us mechanism and hopefully move us forward to translation. We can then take those samples and we can sequence them. So there's lots of different ways we can sequence them, but basically we're saying, okay, who is there? Let's just say, what are the bugs that are in your gut? What are they? And what is their potential function? Because we can do sequencing will tell us all their genes and their potential function. Even better is we can also now send these samples to the Calgary Metabolomics Research Facility, and there we measure the small molecules that these microbes are making that are present in that fecal sample. And then we built, as Derek said, we built the world's largest germ-free facility. Now, why did we do this? Because this is an incredibly powerful system where we can take an animal model, a mouse, and we can raise it with no microbes at all. They live in a bubble, so 0 microbes. And so we can, of course, then look at how does their immune system or any other thing you want to study look like without any microbes present. But more powerful is now we have an empty host and we can give microbes back. So we've developed systems where we can transiently colonize a mouse. We can genetically engineer the bacteria we want to study and make it so that they can't survive very long in that host, so they become germ-free again. And we've used this for questions like, how much does the maternal microbiome influence the development of the immune system in a baby? So we can give it while that mouse is pregnant and she becomes germ-free again before she gives birth to her germ-free pups. And then we can look at the immune system. And now we know that those signals are coming from the maternal microbiome rather than the baby's microbiome. But we can also put in any bug we want by itself. We can put in 2 at a time, 3 at a time, 10, 20, 40 at a time. And that's a notobiotic model. And that just means that we're putting in exactly the bacteria that we know. It means known biota. And so we can do that. So we can say, if we look and we find a microbe that is in a cancer patient that's not responding, we can isolate that, put it into this animal model and say, well, why is that we're not allowing it to respond? And our newest venture is basically, we built a wild microbiome facility. And the reason we did that is that mice that have been associated with a wild microbiome that is much more diverse that we have in our animal models have a better educated and more mature immune system. And so they respond to therapies more like humans. So now here, microbiome research in Calgary, I think we are really leading microbiome research. I think we are considered really the #1 microbiome research group or team in Canada. And we're recognized globally as really leaders in microbiome. And since we launched the International Microbiome Center, our publications in microbiome have steadily increased, and not just in number, but our researchers are publishing in really the highest scientific journals. And we're studying a whole variety of different things. So early life, homeostasis, the gut immune system, the response to infections, the gut-brain axis, cancer, the lung microbiome, inflammation, and also those bacteriophages that infect the bacteria. So last 2 slides. What now? So we really have done super well in the last 10 years. And I really have to say that we would not be where we are now without massive support from our community. We do pretty well getting grants. It's a very competitive field, but there's never enough money in grants to run programs like this. And so, our community has really helped us to get us where we are today. And so what's next? We've made amazing discoveries. I think nobody will argue now that the microbiome does not have a pervasive effect on the immune system and on your health. And now what we need to do is we need to harness the power of the microbiome. We need to translate our discoveries. We need to bring the microbiome to the clinic, to the hospitals, to our communities. We need to use this to have better patient care. And so our dream and our focus now is to build the first and only Canadian Centre for Microbial Therapeutics. And so our vision for this is that it's going to have a couple arms. One is a research and development suite, where we are going to be able to culture and isolate those really important microbes from healthy individuals, characterize them, grow them, validate them through all of our different model systems, and then bank them for use. The other is to build a GMP facility so we can produce these microbes. And that's very important because we need to do this under regulations that are approved by Health Canada or the FDA. And we want to build these precision microbial therapeutics and get it into patients and translate our discoveries. And I'm going to stop there. And thank you. And just say we're super excited for our next adventure. And hopefully you'll be there with us. Thank you.
Thank you, Kathy. I've heard the story a number of times and it just gets better every time. It's phenomenal what you and the team are doing. It's now my pleasure to introduce Dr. Braedon McDonald. Braedon did his PhD here, then he went into medicine, and he did his medicine here in Calgary, then went on to faculty, and he's now an associate professor in the Department of Critical Care. He's a clinician scientist, an emerging leader in infectious disease, and works in the ICU. He's going to tell us about some of his research and most recently, the Snyder Chair in Critical Care. So congratulations on that. The podium is yours.
Thank you, Derek. Thank you, Kathy, for introducing everything. What a great turnout. I'm glad everybody seems to be as excited as I am about the microbiome. So I'm going to, instead of giving an overview, I'm going to work off of what Kathy's introduced and give an example. Okay. It's like Kathy said, we want to bring these discoveries that are fantastic scientific developments out of the lab into the real world. Okay. And I'm going to give you an example of how we want to do this and how we are doing this in the intensive care. Okay. Why the intensive care? Well, because I think it's an area where we have an opportunity to make a huge impact with microbiome focused care. I'm an intensive care doctor. I work in the intensive care that's down the hall in the Foothills Hospital. What is intensive care? It's a place where we provide life support therapy. And I look after everybody and anybody, whether they have neurologic diseases, heart disease, lung disease, bowel disease. The unifying factor is when people find themselves so unwell that their organs are failing and they need life support, they come to us in the intensive care. Okay? And in addition to life support being a unifying factor, the other major unifying factor that I see in my patients in the intensive care is a risk of infections. Okay? So really what I'm talking about today is infections. And this is truly a major problem. And it's a major problem that I face and that my patients face every single day. And these infections are very severe. Okay, we're not talking about head colds and the sniffles. We're talking about deadly bacterial infections that are affecting people who are hanging on for dear life on life support machines. Okay, and you can see just by this graph the percent mortality of patients in the intensive care, and this orange line, you can see how high it is. These are the patients who develop infections. So there's a tremendous vulnerability when your defenses are down, when your systems are under such severe stress. And this opens the door for infections that are deadly. And it can get to a point where sadly, it's almost a flip of a coin, whether my patients are going to get home to their families or not, if they develop these severe infections. So preventing infections, treating infections, that's great. We have antibiotics, and for the most part, we have good antibiotics, but what I really want to do is prevent these infections. And I think this can save lives, as you can see from this data. But also, even in those folks that fortunately survive these terrible infections, there can be long-term consequences that can affect the rest of their lives. So, as I say, prevention is the best treatment. And this is an example of someone who was treated down the hall here in the Foothills ICU. Her name's Verna. You can read about her. There's a really nice article in Reader's Digest Canada about her from a couple years ago. And she's a very healthy, vibrant lady who unfortunately developed a severe infection in her bloodstream. Her organs shut down. She had to receive treatment in the intensive care with multiple types of life support machines. She was in hospital for months. And this really took a toll on her body. She actually lost a number of limbs to amputation as a consequence of the infection. and it affected her organ function long term. And I bring up examples like this lady, her name's Verna, for a couple of reasons. One is, again, to illustrate the gravity of the problem, but also I like to highlight the fact that, you know, even coming through significant things like this, we've got great stories of survivors who go on to do wonderful things. And Verna's an athlete. She participates in lots of different sports, and she's a very active participant in infection advocacy and sepsis advocacy in Canada and around the world. And so if you ever are reading in this area, I'm sure Verna's name will come up, because she's a really vibrant lady who wants us to do better for the next people that find themselves in the ICU. And there's other examples. You can read about them all the time in the news, but they might fly under your radar. This is an example from just this summer. It was in the media of this young fella. His name's Ben. He was an Olympic wrestler and a UFC fighter who, despite being young and in the peak of health, developed a severe staph infection in his lungs, a pneumonia that landed him in the intensive care. And just a few weeks later, you can see the toll that it's taken on his body. And he survived, but he had to have a double lung transplantation because the infection was so severe. And he made it out of hospital, but still, I saw in the news just a week or two ago that susceptibility to infections lasts, and Ben's back in hospital again with another infection. And so this is the problem that I think we can tackle, because these infections are from the microbiome. When your defenses are down, when your system's vulnerable, these bugs that in health are so helpful and so critical to our well-being, well, the bad ones can take over when your defenses are down. And in the gut and other sites like Kathy described, those good bugs are outcompeted by the bad ones, and they can get out into the bloodstream and in other organs and cause these terrible life-threatening infections. And so in our lab, and we work closely with Kathy and other collaborators here, we've done research in the laboratory to identify who are these bad bugs. Why are they becoming bad? Why are they misbehaving? What are they doing to us that's causing these infections? And we can take it further now, and we've developed ways to correct this. We have good findings, including using the resources in the IMC with these mouse models, that we can cure these problems in mice, and we can reverse some of the damage that's done to the microbiome and the immune system. And this is really exciting. But like we're talking about here, research and these discoveries, they're fascinating, they're motivating, but at the end of the day, it's about bringing it to the people who need it. And in my case, it's my patients in the intensive care. And what we can accomplish in the lab, we can't always accomplish in the real world, because the laboratory is a highly specialized, highly uniquely structured environment that allows us to do very complex things that often just aren't feasible. And so we can make these fantastic microbiome discoveries based on averages from hundreds of patients. We identify signals, we develop potential cures. But when I'm standing at the bedside in the intensive care, I need to be able to apply these findings to my patient, who's an individual. He's not an average. This is an individual person who deserves individualized care. And that's the challenge, is bringing this research out of the laboratory into the real world. And I say challenge accepted. And that's what we've been working on for the last little while, is how do we understand the microbiome in the real world? And that's required a little bit of elbow grease and technology development, but I think we're getting there. And that's what I'll tell you about now. So I want to be able to understand an individual's microbiome in the ICU. And we need to be able to do that in a way that's fast, because minutes and hours count in my world. And it has to be cost effective for our healthcare system to be able to use this. And I need to be able to apply it when I need it. And so we turn to this machine. No, not that one, this one. Okay. So this is a DNA sequencer that lets us analyze the microbiome. And this is in the lab, just across the wall over there. But you can imagine this is not very feasible in the intensive care. We need something that's small, portable, accessible, cheap, and that this little guy is what we turn to. And the reason we turned to that was because a couple of years ago, I was reading an article that they'd taken a DNA sequencer up to the International Space Station and were doing DNA sequencing. I'm thinking to myself, how in the world did they get this onto the International Space Station? But no, in fact, it's this little gizmo that's just smaller than a cell phone that you can plug into a laptop computer and you can do complete DNA sequencing with. So we took this technology and we began working with it and adapting protocols and adapting ways to analyze the microbiome with it. And to cut a very long story short, you know, this work was really propelled, again, as Kathy said, by our partnership with community. It was supported by a Snyder Institute granted Catalyst grant, where the intention was to try and move discoveries at lightning speed towards clinical care. And what we did was we developed a protocol where I can take a sample from one of my patients in the intensive care unit, and within a matter of hours, we can isolate DNA, do full microbiome sequencing, full computational analysis, and I can get results back that are clinically actionable that can tell me, does this patient have a bad microbiome that puts them at risk of infection? And what are its bad features that we need to correct therapeutically? And just to give you an idea of scale, our conventional sequencing method, we can do hundreds of samples at a time, but I need to do one on my patient, and it needs to be fast. This sequencing method takes weeks to months to get results back. That's no good. And per patient test, so one test would cost upwards of $4,000 to run. With the workflow that we've developed with this little gizmo that you can plug into a laptop computer, we call it On Time, we can get full microbiome profiling within a couple of hours for just over $100 per test. So this actually allows us to bring the microbiome to the bedside so that I can use it to treat my patients. And so that's exactly what we're going to do. And just as Kathy said, we're excited for the next phase, but it's not theoretical. We're doing it. That's our attitude around here, is let's just get to it. And you know, the traditional mechanism to allow us to do this through, you know, government grants and things like that, that's something that we will continue to pursue. But really, it's the support of the community that's allowing us to leapfrog some of that red tape and the barriers that make things take a decade to go from the lab to the bedside. We're trying to shorten that down and minimize it to the best of our ability by partnering with excited people in the community that want to see this come forward for the benefit of our patients. So we're in the process of starting up this clinical trial where we're going to sample the microbiome of my patients in the ICU, use our on-time rapid sequencing to analyze the individual microbiome of that person so that we can provide microbiome-guided infection prophylaxis, so preventative therapies, prevent infections before they start. And again, the goal here is personalization. Patients are individuals. I want to treat people. I don't want to treat diseases. We're taught to treat diseases in medical school, but I want to treat people, and that includes knowing their biology, knowing their microbiome, and treating them in a personalized way. This is all integrated with what Kathy mentioned, which is our goal and our intention to build the Canadian Centre for Microbial Therapeutics, so that in addition to being the leader in the world of microbiome research, we can become the leader in microbiome healthcare.
Okay, now comes the fun part. And by fun, I mean fun for me because I don't have to answer anything, and fun for these guys because they do. Thank you for coming today. I'm Craig Jenny. I'm Deputy Director of the Institute, and today I'm the referee for making these people suffer. And as somebody who doesn't work in a microbiome, I mean, this to me is still just enthralling science. I mean, I recall how, I don't want to say how many decades ago, But when I was a student, the concept of treating diseases with antibodies seemed like science fiction, and now it's everyday science. And here we are on the step of the next big advancement. So very exciting stuff. So we have two question formats. You can upload the questions digitally, or we have some microphones floating around. So if there's questions, put up your hand, a mic will find you. But maybe we'll start here with something online while we sort out our first person. And the first question, let's put it maybe to Dr. McCoy here. Is it possible to re-educate the microbiome in autoimmune diseases?
Yeah, I mean, we're not there yet, but we certainly hope so. So my lab does have a project where we've been studying the microbiome in type 1 diabetes. And to re-educate the microbiome, we want to change its maybe composition or its diversity at the right time. And so we don't necessarily know in autoimmune diseases, maybe in specific ones, what is the best microbiome to have. And remember, when we hope that we could give a microbial therapeutics, a defined consortia population of bacteria that is going to help that person. But we also have to think about maintaining that microbiome. And so we have to think about the diet that the person is going to take, that the person is eating, and how far along their diseases and their health. I'm an optimist. I would say yes, we hope that we can re-educate the microbiome to help to have better, symptoms, better control of your autoimmune disease. I don't think we're there yet, but hopefully soon.
Thank you. And I think we'll go with the room here.
Question for Dr. McCoy. You mentioned antibiotic history affects the microbiome, and I get that would be the case. But my question is it better, worse, or just different?
Yeah, that's a really good question. So if you think about, people take antibiotics. You hope people take antibiotics because they need them, that they have an infection and you're really trying to target a pathogen. But antibiotics, especially a broad spectrum antibiotic, is like an atomic bomb to your commensal microbiome. And so there's been studies that have been done to show that you can lose microbial species from taking antibiotics, and it's gone. It's gone from your microbiome. So what happens is after over and over and over antibiotic use, you start to lose the diversity. So the number of microbes that are there. And the idea is that when you lose diversity in microbiome, your microbiome becomes less resilient. And so when you have a low diversity microbiome, it's not very resilient. So when it gets a hit, like another antibiotics or some other thing that challenges it, it's not able to weather the storm so much because you have many different bacteria that are in there that maybe can do similar function. But you start to whittle that away when you lose diversity. So, you hope you target the pathogen and you get rid of that pathogen, but you always are going to have collateral damage on your commensal microbiome.
Great. We'll go maybe back online here now, perhaps to Dr. McDonald, and I'll modify this one a little bit. Can I walk into a clinic and request a microbiome sequencing? Maybe I'll add yet and or follow it up as to when.
Very good question. So there are a very small number of very specialized private clinics. I think there's one or two in Calgary and a couple others in Canada where you can go and request a microbiome sequencing. They'll take a sample. Usually it'll be a poop sample. And in a couple of weeks or sometimes months, you'll get a report back. Whether that report is just all Greek to you or has any sort of interpretation, tough to say. I've never personally done it, and I've not seen these reports because they don't go into the public system. So the short answer is yes, but to what end? Because at this point, these data, unless they're properly interpreted, unless we know what to make of it, And frankly, we have something that we can do about it if there's a problem. It's simply information. And in the vast majority of medical settings, the answer is no, for the same reason. Because while the technology is there in the research realm, it hasn't yet been pushed forward to a point where we can action it. And so that's what we're working on doing. We're working on doing it in a way that we can democratize access to this so that anyone who needs it can have access to it, and that it can be supported in our fantastic publicly funded healthcare system in a manner that's cost effective, but that it's also always linked with, well, if we identify a problem, what are we going to do about it? How are we going to fix it? So that's a long-winded answer. It's kind of a waffling answer, but the complexities are there. So yes. if your credit card has a
That's right.
fairly
Yeah,
high
yeah,
limits
yeah.
on it, I think
It's
you can
yes.
get microbiomes.
Yeah, I think we'll go to the room here.
Are you hoping that we can like also introduce this kind of microbiome treatment into like other like places other than the ICU, like maybe cancer research or like cardiology or something like that?
Excellent. So I give you the example of the ICU as an example. It's one area that's near and dear to my heart because that's what I do every day and lots of the nights as well. But the nice thing about microbiome therapy as an avenue to really propel healthcare is, as Kathy and I often say, it's disease agnostic, meaning that it really, we're accumulating evidence that the microbiome is implicated in almost every disease condition, directly or indirectly. And that includes neurologic disease, heart disease, lung disease, valve disease, everything, acute and chronic. And so are we hoping to implement this? We're going to. We don't just hope. That's what we're going to do. Because I think lots of people stand to benefit from this. And we don't want to restrict it to just one area like the intensive care. We want to, again, democratize it so that it's available to everybody that needs it.
Thank you. Dr. McCoy, I'm going to try awkwardly to mash 2 or 3 points together here on screen is moving faster than I keep up. So good work, but probiotics. Do they help rebuild your microbiome? Do they simply protect the microbiome you have? And if you are going to be prescribed antibiotic, particularly, a strong regime, should you be requesting a probiotic prescription with that?
So it's a really good question, and it's a bit of an emotive area. And there's some strong
welcome.
opinions about it. And for me, I struggle a little bit with the word probiotic. And the reason is because it's traditionally, the probiotic has been like a beneficial microbe that has been heavily marketed, not with any basis in science. And so, there's a lot of marketing hype about probiotics. And traditionally, you can buy a lot of probiotics on the shelf that you can take, and many of those are no longer alive. There was one study that pulled a bunch of probiotics off the shelf and sequenced them, and many of them were actually isolated from chickens, and so they're not really from humans. So it has a bad rap in the past. But as microbiome research has developed and evolved over the past 10 years, there's been a lot more of a push on doing better science with probiotics. And now there's been better studies and better clinical trials, better design trials. But honestly, the majority of them have not shown the benefit. And that may be because they chose the wrong probiotic, but maybe it wasn't really rationally designed approach. Maybe it was the wrong probiotic for the wrong individual, for the wrong disease. I think that there is some evidence, especially in post-infectious diarrhea, that that giving probiotics within 2 days of that has shown in a trial benefit. So I try to never use the word probiotic. I try to use the word microbial therapeutics or microbial therapies because I think that distinguishes it from that it is based in scientific backing and rationale for the approach. But there's probably many incidences and, you know, anecdotal, like different people talking that took probiotics and may help. But overall, the clinical trials that have been done are just maybe not designed right. And overall, there hasn't been that much benefit. Do you want to say something?
My father had to go on antibiotics last year, and I had him put on probiotics so that he wouldn't have diarrhea after the antibiotics.
We're gonna go for a live question here.
Thank you for your amazing talk. Will there be a possibility of having a microbiome transfer center, like there is a blood donation center?
That's a very, very interesting question.
Yeah.
That's a really interesting question. So the field of fecal microbiome transplantation is very interesting for many reasons, including the fact that actually the number of people that serve as donors for microbiome transplants is very, very small. And to give you an idea of scale, Calgary's a little bit different, but when I was a medical resident in Vancouver, for the entire metropolitan Vancouver area, the whole fecal transplant program, which is big that they run there, was supported by one single donor, one person who just pooped in a bucket all the time and brought it in for them to make. So, but what's
There's
the reason for that?
Vancouver for you.
Yeah, it's because that's right, exactly. Eye rolls, right? So one of the reasons for that is currently the state of science is that there is huge scrutiny on who can be a donor for fecal microbiome transplants because we're learning so much about how the microbiome is implicated in different diseases. There's a concern that if that donor has a problem, it could be transferred to somebody else. And so there's a huge long list of exclusions for people being fecal transplant donors, including mental health diseases, including skin disorders, including previous cancers, including family histories of things, and a huge long list. And so even though there might be a lot of people motivated to donate, it gets whittled down to very few who actually qualify. But I suspect that as we learn more, we gain more understanding of what can and can't be transferred by fecal transplant, that will be opened up again in time. I'm not sure to the extent of like a blood bank system, but then just to put in a plug for, you know, the development of precision therapeutics, it takes away those concerns when instead of just using the poop from somebody else to transplant into an individual, you've actually engineered in the lab a cocktail of microbes that specifically targets the problem. And then you're not worried about the transmission of things from one person to another.
Robo poop.
Can I add to that? So the other thing about fecal microbial transplantation and the donor is, first of all, not every donor is equal. So in Clostridium difficile infection, the donor doesn't seem to really matter because this is used to basically repopulate the microbiome that has been taken over by Clostridium difficile. And so it's a kind of a low bar that you have to pass. But the other diseases that we're trying to look at have a bit more efficacy that needs to be looked at. And many FMT studies have really showing a donor effect. So a study in McMaster and IBD, they had some really promising results in FMT, but it was from one donor. The other donors had no effect. And so they had a super donor. And the problem is I heard, not officially, that they lost their super donor because the super donor got an infection, had to take antibiotics. And after that, they were no longer superdoning. That cancer study I told you about where they did FMTs in those melanoma patients that had failed therapy, one of the studies used two different donors. One donor had no effect at all, and the other donor was the only one that actually had a complete response and partial response, telling us there's donor variability. And, we're using that information to try to help us figure out why. But using this more precision microbial therapeutics, we're going to get away from that because we're just going to make these microbial consortia based on what we know is working and not just hoping that one of you are going to give you proof and one of you is going to be superdonor. And stay that way forever.
Yeah, that was a great question because I think we took 2 or 3 off the list here. So we've got melanoma covered now. There was one on the list. I think it's off the screen, but I'll throw back to Dr. McDonald. It comes from this idea of Port Vancouver, and I think it's because it being a Canucks fan also was one of the pre-existing. There was a question about how can people in Calgary find out what other clinical trials are happening, whether it be FMT or others. How can they participate in clinical research? I know that's a question that comes up a lot. So, with no prep, here you go.
Absolutely. So in some fields, access to clinical trials has been very well coordinated, and there's good forward-facing public presence to this research. You can go on the Crohn's and Colitis Foundation of Canada website or the Heart Association website, and you can learn about what's going on and what you might have access to in your area or other places that you might be able to go in order to participate. The microbiome field, it's so new that hasn't been developed. These organizations are decades old. They've had a long time to build that infrastructure. But, by bringing together researchers, clinicians, and folks that are pushing things forward into clinical trials through an entity like the Canadian Centre for Microbial Therapeutics, I would envision that we could finally start to consolidate this in a way that the public would have access to information around what clinical trials are going on. Would I be eligible? Who should I contact if I want to participate? And then once we've moved forward out of clinical trials, what's been approved for my health problem and where can I access it? It starts by building a system, and that system doesn't exist currently, but that's what we're motivated to build. And we want Calgary to be the epicenter of that because I think we're ready to lead that drive. And so it's not there yet. So I can't tell you yet where to go for that information, but give us time and we'll get there.
Thank you. So got about 5 minutes left. We'll go to another live one if there was. Nope. Yep. Right behind you is the.
For hosting this tonight and putting this on. Looks like a very variable problem here that you're working hard towards. My question is this: does data exist to show changes or trends in human microbiome over any period of time? And can you attribute any causes to that and any effects of that?
So big question. And really it depends from what angle you look at. So we can, if we think about it on a global perspective, we were fortunate just this morning to host, I would argue, the world's leader from the Sanger Institute in UK here, just in the other conference room, who talked about global variation in the microbiome. And the fact that, in fact, microbiomes in industrialized countries are very different than microbiomes in non-industrialized countries. And that's likely driven by many factors: diet, other environmental features, environmental exposures to antibiotics and everything like that. But when we're talking about a big picture like that, it is very correlation based. But when we start drilling down to specific conditions where we can actually start answering 1 + 1 = 2 type of questions, absolutely we can see evidence that particular exposures lead to changes in the microbiome that are detrimental to health, which when restored are curative. And by far the best and most clear example of that is C. diff infection, which is most commonly occurring after somebody's taken powerful antibiotics and the microbiome's been decimated, opening the door for C. diff to come in and take over. And yes, antibiotics to try and kill C. diff are effective in some people, but if they're not, the cure is repopulating the microbiome with fecal transplant to cure the disease. So that's the best example that we have, the most crystal clear to show direct evidence that these shifts are disease causing, that restoring the microbiome can cure. But now we're gaining very similar evidence in a variety of other diseases like Kathy mentioned. And so I would say the answer to the question is yes, but the granular details really are in specific patient populations with specific diseases where we can measure things granularly rather than, big picture things, on a population level or global level. Does that answer your question?
Gastrointestinal cancers on the rise.
Yeah.
You hear about, I read the other day, appendectomal cancers on the rise. Things like I had a conversation with my doctor, shingles in younger people, including myself, a couple of years ago, are on the rise. This started within the last decade or two. These trends have emerged. In your presentation here, you say that so many of these things can possibly be linked to microbiome changes. And this is what I'm saying is like, have the correlating factors been identified in some of these things?
Yeah, so there's a hygiene hypothesis. I'll turn it over.
So the general way of thinking that we believe now is that, so we believe that since in the last sort of 60 years, it's really, or slightly longer, it's really clear that there's a huge increase in chronic disease. And that is allergy, so allergic disease, asthma, atopy, food allergy, autoimmune diseases like type 1 diabetes, now metabolic disease, that there's a massive increase in this over time. And the idea is, our genes in humans don't change over that short period of time, over, the 60, 70 years. And that it is really that all of these diseases are due to a gene environment interaction. And so what has changed? If it's not our genes, it's our environment. And the hygiene hypothesis suggests that as we have become more industrialized and people have gone away from from rural living and lived more in cities, we've changed our diet massively. We way more processed foods, all of these things, and that we are not exposed to microbes like we were in the past. That all of this increase in hygiene has caused this massive increase in these immune-mediated diseases or chronic inflammation. Now, our field, so that hygiene hypothesis is really like, if you live in a family with a variety of kids, the youngest kid is the most protected because the other kids bring microbes home from school and have more interactions. They bring it home, and so the youngest kid, larger the family, the more protected the younger ones are. If you live on a farm, you're more protected from allergic disease and developing allergies later in life. That's all part of this hygiene hypothesis. Now, as a microbiome researcher, because we know that the microbiome is hugely impacted by the environment and about things that we do, our diet, our exposure to farm life, or how much processed food that we eat. We think that the microbiome is sitting at the interface between that and outside environment and our inside body, and it's the real stuff. It's changing in response to the environment, and we're losing diversity. When we lose diversity, we start to lose some of these key signals that your body has co-evolved to expect. So we've co-evolved with our microbes. And so we need some of these microbial signals during early life development to develop your immune system in the right way, to make sure it's developing a regulated way. And so That's the whole where the field is now. And the idea is that after generations living in westernized, industrialized countries, you know, the first generation had a hit and lost some diversity of the microbiome. That's then vertically transmitted from the mom to the baby, and then they live in that environment. That generation gets another hit. And after several generations, you start to get this really loss of diversity in the microbiome. And then you don't have this resilient population anymore. You don't have the diversity to give you all those signals that you need. And there's a lot of correlative, associated studies in humans that will really back that up. And then a lot of research in animal models that will back that up. So we believe that changes in the microbiome are really impactful. Now, I always get the question in microbiome, you know, if microbiome is associated with a particular disease, you know, say, I don't know, IBD, is it causal? Did you have a change in the microbiome and that caused my disease? Or is it just a consequence? Because we can sequence the microbiome and nearly every person that has a disease, it's going to look different to a healthy individual. So, but is that just an association? It changes because there's a disease? That's a really difficult question to answer in humans. But what I would try to say is the microbiome is always complicit. So whether it's causal, maybe you had a change and it was one of the key driving factors that led to the disease. But when you're diseased, your microbiome does change. It changes under inflammation. It changes because your diet changes. It changes about all kinds of things. And it doesn't usually change for the better. So like Braedon said in the ICU, their microbiomes are changing like crazy. They're in a terrible situation. And, you know, they're fighting for their life. Their microbiome is changing. And it is no longer resilient. They're all on antibiotics. So it's definitely complicit. If you have a disease and more inflammation you have, your microbiome changes, and it's probably driving more disease. So I hope that helps you.
Yeah, unfortunately, I think we're going to have to wrap it here. We still have more than 30 questions online, and I see a couple more in the audience, but we've blown well past our scheduled time, which is my fault. So with that, I'd like to thank our presenters today, and please. And would like to invite everybody downstairs. Oh, the boss has a few things to say, so I'll let the boss go here.
Thank you, Craig. Yeah, I hope you've all heard and seen the passion and the quality of Dr. McCoy and Dr. McDonald. And these are two of many of our researchers. And that's been the mantra over the last 15 years or so at the Snyder Institute is recruit the best talent, provide them with world-class infrastructure, and then, as Braedon said, get to it. All right. And that's exactly what's going on. So thank you very much for tonight. That was fantastic. So in closing out, I only have a couple of things just to round up here. So first of all, thank you for attending in person, and thank you who are online for attending and the very insightful questions. For those online, have you additional questions? If you'd like to reach out, you can get us through the website. Please don't be shy. Reach out and contact with us. And I really hope that you were impressed and inspired by this health-based research in the Snyder Institute that will undoubtedly improve the lives of individuals suffering from chronic and infectious disease. The Snyder Institute is on a remarkable trajectory upwards in improving healthcare. And you, our community, have been really critical in making this a success. We invite you to connect with us, share your ideas, the challenges, and the questions. Defeating chronic disease is really a team effort, and we need your input and we need your support. You're part of the team. Our researchers are highly competitive in attracting peer-reviewed grants. But as we've all told you tonight, this is a long process. Really excellent, outstanding grants can take 2 or 3 years to get funded. And that's really a reflection of the budget going into research. And that's where our partnership with the community is so helpful. Your support moves things quicker, faster, getting from the bench to the bedside, really making a difference in the lives of individuals that need that cutting-edge treatment. So giving to the Snyder Institute speeds up the discovery and the translational research. And we are in the process of defining new microbial medicines, living medicines, microbial therapeutics. And the community support, your support helps leverage additional grant support. The support from the community allows seeds grants, allows more research, data. That data makes for a much stronger grant to the federal or provincial government, and our success spirals upwards. So if tonight's conversation and data and directions has resonated with you, I invite you to reach out to Megan Robinson. Megan is over here. We'll show you here. And she's our Director of Development, and we'll be very happy to tell you how we work, listen to your comments, and show you and how you can help support this really exciting and innovative initiative. We have some cards downstairs, and her contact information is on that. So if you don't have time to talk to her tonight, you can contact her through our website or an email. So finally, please join us in the conversation. It's important that we hear from our community. We are really indebted to the community support. We're going to have a little reception downstairs. Everyone is invited. A number of our researchers will be downstairs, including Braedon and Kathy. If you look on their badge, there's a little green dot. Really have a chat. Ask them what they do. You'll hear the excitement. You'll get excited. We want to engage you. Thank you for coming tonight. It's been a real pleasure.